Hello, my name is Valerie Lantigua, and today I'm going to be talking to you about how mammary gland cells respond to changes in endocrine signaling caused by heat stress during the dry period. So for a little bit of background, heat stress in dairy cattle is detrimental to their health and productivity. As global temperatures continue to rise, understanding heat stress and implementing heat stress abatement becomes increasingly important for dairy farmers. Previous studies from our group have shown that heat stress is associated with altered hormonal responses in the mammary gland during the dry period. For a little more background, the dry period is the time where the cow is no longer being milked before having her calf. This period is critical to the productivity of the cow, as during this time, the mammary gland goes through a process known as involution, where old worn-out cells are replaced with new ones that are primed for milk synthesis. Our hypothesis is that disturbances in prolactin and estrogen in heat-stressed cows during the dry period is what's responsible for regulating mammary cellular processes, such as apoptosis, autophagy, and cellular proliferation. Moving on to methods, let me start off with our experimental design. Approximately 46 days prior to calving, our cows were separated into two treatments, a cooled and a heat-stressed group. Both groups were housed in shaded barns, but the cooled cows had access to fans and soakers while the heat-stressed group did not. Respiration rates and body temperatures were measured from the cows in these two groups so that we could verify that our treatment was affecting the environment of the cows. Throughout the dry period, biopsies were taken from the mammary gland of a subset of cows from each group for ex vivo experimentation. These tissues were then cultured in a media containing the hormones estradiol and prolactin in concentrations that were meant to mimic the average circulating levels of these same hormones in cooled and heat-stressed cows respectively. The plates were then incubated for 24 hours and ran through an LDH assay to verify cell viability. Transitioning into my results, what we found is that the body temperatures did indeed trend higher throughout the day in our heat-stress group when compared to cooled, indicating that the fans and soakers were successful in thermoregulating our cooled group. Figure 6 represents my data on tissue viability after being incubated for 24 hours in the media, confirming that our in vitro protocol was appropriate for the tissue and that these cells will be able to serve as a suitable experimental model in future work. Figure 7 shows the regulation of genes in heat-stressed cows when compared to cooled cows, and what we're seeing here is that the genes relating to apoptosis, autophagy, and cellular proliferation are downregulated at day 3 relative to dry-off, but then upregulated at day 35, indicating that our heat-stress group is having a delayed involution response when compared to our cooled group. This likely means that the following lactation will be negatively impacted. We are still currently analyzing how our ex vivo treatment through the media affected the responses in these tissues, but are excited to see what we find. Thank you.